Hello, good morning, everyone. I'm a PhD student from Queen's University, Belfast, and today I will be presenting some of my PhD results. And so the title of my talk is Enhanced Efficacy of Stimuli Responsive Liposomes of a Doxorubicin PSA Cleavable Prodrug for advanced prostate cancer. And just to give you a very brief overview on the disease, prostate cancer is the most common cancer in men in the UK and is the second leading cause of cancer-related deaths. It is a very complex disease which involves different stages, and therapies will also vary according to the stage of the disease, but our research efforts will focus on the latest stage, or advanced prostate cancer, because this is currently a true clinical challenge for which there are no effective therapies available. In terms of prostate cancer diagnosis, prostate-specific antigen, or PSA, is a serine protease that is elevated in prostate cancer and is currently the gold standard for prostate cancer detection. But our main goal is to use not as a diagnostic, to use the high levels of PSA not as a diagnostic tool, but rather as a therapeutic target. And so the central piece of this project is a prodrug of doxorubicin that is cleavable by PSA. This prodrug, which I will be calling Dox PSA throughout my talk, was initially developed by Merck with the aim of reducing doxorubicin-associated cardiotoxicity. But this prodrug did not go beyond phase 1 clinical trials due to toxicity concerns. And so the aim of this project is to improve the therapeutic efficacy of Dox PSA prodrug in advanced prostate cancer. And to achieve this, we need first to understand the mechanism behind Dox PSA toxicity, and second, to improve the prodrug solubility and stability, as our ultimate goal is to reach an in vivo stage where we need a prodrug with favorable pharmacokinetics and biodistribution. So with these objectives in mind, we thought of using stimuli responsive liposomes, which are able to encapsulate and protect the prodrug from premature degradation in the bloodstream. So the first thing we did was to confirm the high levels of PSA expression across different prostate cancer cell lines. And as you can see here, by real-time PCR, C4-B cells, which represent advanced stage prostate cancer, indeed express high levels of this enzyme. These results were also confirmed by Western blots and ELISA, where we could observe that, again, C4-B cell lysates and also cell supernatants overexpress PSA in comparison to LNCAP cells. And so the next thing we did was to test if the prodrug that we synthesized in our lab was actually active and selective towards PSA-positive cells. And for that, we treated both PSA-positive and also PSA-negative cells with either doxorubicin, that we used here as a positive control, or the Dox PSA prodrug. And as expected, doxorubicin was not selective. The prodrug was selectively toxic to the cells that secrete this enzyme. So this was a good starting point to the project, but we wanted to know more about the mechanism of action of this prodrug. And so what we did was we performed some confocal imaging to look at the cellular uptake of this prodrug, and we rely on the intrinsic fluorescent properties of the prodrug to easily track the intracellular localization. And what we observed immediately was that whereas doxorubicin has a very fast nuclear uptake, the prodrug internalization is delayed. And to understand why this was happening, we performed some co-localization studies, and we observed that whereas doxorubicin enters the cells mostly by passive diffusion, the prodrug is internalized via endocytosis, and this is shown here as by the perfect overlap between the green fluorescent signal of the endosomal compartments and the red fluorescence of the prodrug. But what was more interesting for us, was to observe that after 24 hours, the prodrug was able to be translocated from the cytoplasm of the cells to the nucleus. And this was a very important observation because it shows that the prodrug can be activated intracellularly. So to confirm these results, we performed similar experiments in PC3 cells that do not express this protein, and we could not observe any nuclear uptake, as you can appreciate from these magnified regions, where we can see that the prodrug is mainly localized in the perinuclear region of the cells. And just as a proof of concept that the prodrug by itself does not bind to DNA, we performed some drug DNA binding studies. And as you can see here, for a fixed amount of doxorubicin, as we increase the concentration of DNA, the absorbance peak of doxorubicin progressively decreases, but the same was not verified with the prodrug. And so we concluded that the DOX PSA does not interact with the DNA, and so the nuclear localization that we observed in PSA positive cells was a result of the prodrug being hydrolyzed by PSA and then entered the nucleus of T cells as free doxorubicin. So now what happens if we encapsulate this prodrug inside liposomes? And to ensure that we have a fast and efficient release of the prodrug, and this is very important because we need DOX PSA to be accessible to PSA hydrolysis, we used low temperature sensitive liposomes that rely on an external stimulus by temperatures around 42 degrees C to release the prodrug. And after troubleshooting through different drug loading techniques, we selected the one that provided the highest encapsulation efficiency, which was remote loading. And here I just want to point out that the drug loading was very low compared to the usually obtained drug loading for doxorubicin by remote loading, that is above 90%, and this shows that having a molecule of doxorubicin conjugated to a small 7 amino acid PSA cleavable peptide completely changes the kinetics of encapsulation and diffusion across the lipid bilayer. Here we have some cryoem images of the empty liposomes on the left, and also the prodrug-loaded liposomes on the right. And we can nicely see the vesicular structures of these liposomes, but we could not conclude on the drug loading, probably due to the lower encapsulation efficiency. So to have a more detailed view of the structure and specifically the core of these liposomes, we performed freeze fracture technique, and here we can see, as pointed by the white arrows, some drug crystals in the core of these liposomes that were not present in the empty liposomes. So the next question that we wanted to answer was if the prodrug could be released from these liposomes. And again, we looked at the cellular uptake, and as you can see here, after 24 and 48 hours, the free prodrug is already localized in the nucleus of PSA positive cells. But the same was not verified for the temperature-sensitive liposomes, where after 48 hours, the prodrug is still in the cytosol of the cells. This is not surprising because these experiments were performed at 37 degrees, so physiological temperatures, where these liposomes should be stable. But what was very exciting for us to observe was that when we apply hyperthermia, there is a quick release of the prodrug from these liposomes, although it doesn't reach 100%. And after 48 hours, we can detect the prodrug in the nucleus of the cells. So then we moved to assess the cytotoxicity of this formulation, and for that we used PSA positive, so the C4-B cells. And we used the free prodrug as a control here in gray. And then once we apply hyperthermia, as we can see by the red bars, there was a dramatic decrease in the cell viability. So this was very promising results. And just before moving to the in vivo stage, we wanted to use a more in vitro representative model of the tumor microenvironment. And so we used 3D prostate cancer spheroids. I must say that they are not as complex as the ones that Professor Mura just presented, because it's just the prostate cancer cells, but if it's the purpose, we want to assess more the drug permeation across these spheroids. And these C4-2b spheroids are very, very compact. So it was a good model also to assess if the drug and the liposomes penetrate well. And what we could observe also a very significant decrease in the cell viability of the spheroids after applying hyperthermia, and more importantly, the temperature-sensitive liposomes in combination with hyperthermia were even more effective than the free prodrug. So this was very promising results, and the next stage is really to test this formulation in vivo. And just some concluding remarks. So we provided a better understanding of the mechanism behind DOX PSA toxicity. We performed cytotoxicity, cellular uptake, and drug DNA binding studies to show evidence of prodrug selectivity. We were able to encapsulate the prodrug inside temperature-sensitive liposomes, and we showed that the prodrug could be released upon mild hyperthermia, and that the toxicity of these liposomes in combination with hyperthermia was superior to the free prodrug, both in 2D and 3D models. So the next stage, as I just said, and actually this is where we stand now, we are doing some in vivo testing to show if this formulation is indeed more efficient than the free prodrug. And just to finalize, I would like to thank CLINAM Foundation for the opportunity to be here today to present my work. Also to my PhD supervisor, to all the members of my research group, and to the collaborators for the cryoem and freeze fracture images, to all the funding bodies, and to you for listening.
Thank you very much. Questions? Yes, please.
Thank you very much for this excellent talk. This is very exciting. I have never heard of temperature-sensitive liposomes, and I saw that you use DMSO. I did not know that it falls apart if you heat it up to 42 degrees. Could you comment on that, as well as on how you want to induce hypothermia in the animals?
Sorry, I didn't get the first part of the question.
The first part is, how do you generate temperature-sensitive liposomes with DMSO?
DMSO? No.
You had, on one slide, you were using DMSO?
No, sorry, the DMSO is in the cell viability, and it's just a control of toxicity, I think.
Oh, sorry.
Here.
Oh, okay, okay.
It's just a positive control for the cell toxicity.
I thought it's a vehicle.
No.
Sorry. Yes.
So the temperature-sensitive liposomes is a combination of lipids, but the main aspect is that there is a lysolipid that forms above the phase transition temperature. It forms pores in the bilayer that releases the drug.
Okay, would you be able to tell us which lipid that is?
Is MSPC, is reported, is the Thermodox.
Okay.
That is currently in the phase 3 clinical trials.
Okay, thank you very much.
Any more questions? In the back.
Thanks for the great lecture. I just wanted to ask, like, how did you induce hypothermia? Was it using ultrasound or microwave or?
Yes, so in these studies, I used simply a water bath heated at 42 degrees, and we controlled the temperature with a fiber optic to make sure that it's stable and it is kept at 42 degrees. The in vivo studies, we will be using a similar setting, but obviously in the clinic, it needs to be something more like high-intensity focused ultrasound or radio frequency ablation. This is just a proof of concept of the system that is working with hyperthermia.
Thanks.
Well, I was wondering how you survive 42 degrees in the body when you have to heat up the... When you treat me with these treatments and I have to have high fever of 42 degrees, it will kill me before the therapy really gets going, right?
Actually, in Germany, there is a clinic that is already doing is the whole body hyperthermia. So what I have, they use like thermal blanket
Wow, okay.
that heats. Of course, it needs to be very well controlled, but I think so far none of the patients died.
Because when my baby gets 40 degrees, we get nervous. Any more questions? Go ahead.
Can I have how to activate? Have you thought about in the human, in the male? Is the plan to activate this with brachytherapy, for instance, when you have needles in the prostate where you can heat up the prostate? Is that your objective?
I think
for a localized therapy? No?
Yes, this is already done with the needles in the clinic as well.
Have you tested that, or have anyone tested?
Not yet, not at this stage.
Okay.